Good morning. I'd like to thank the organizers for inviting me here and giving an opportunity to share with you the results of our work. I don't really know in which shape or form my presentation would qualify for the keynote presentation. It's sort of technical and focused, but it seems that's what you are getting anyway. So I'd like to switch gears now. So how do I get to my... So I'd like to switch gears and talk about drug discovery. We change more specifically small molecule drug discovery for influenza. As Dr. Umlauf already mentioned, there are several classes of flu drugs currently on the market. So there is M2 ion channel blockers, and there is a number of neuraminidase inhibitors, including not on the market, but described those which covalently bind to the neuraminidase. So we'll be focusing on non-covalent binders of neuraminidase. And more or less recently, there is a first representatives of the polymerase inhibitor class, which is favipiravir, or better known as T705. This is an old Toyama compound, which is currently on the market in Japan and in phase 3 clinical studies in the US. So oseltamivir, or sold-as Tamiflu, is a bioavailable drug, which is nice, and it's efficacious in vivo in inhibiting influenza virus. Zanamivir is not orally bioavailable. It is administered as through inhalation. And paramivir is only formulated for intravenous injections. Right? So there is only one bioavailable drug here. And the problem with Tamiflu is resistance, which is quickly spreading. So it's not uncommon now in some regions of the globe that more than 99% of circulating viral population is resistant to Tamiflu. Right? So what we thought would be nice to do is to try to develop a neuraminidase inhibitor from the same class, which would be orally bioavailable as Tamiflu and would be effective after oral administration, and at the same time would be hopefully more active, and especially active against Tamiflu-resistant mutations. So we engaged into rational drug design for a change, and we saw that if we just collect the data which are already available, derived from co-crystallization of neuraminidase with neuraminidase small molecule inhibitors, and there is some structural data available. So we thought if we get that and try to develop a pharmacophore model for neuraminidase inhibitors like this, and then try to design analogs which would be somewhat different but yet act through the very same binding to the active site and very same mode. So that's a covalently bound compound which was published. So it was determined that through structural studies that it actually makes a covalent bond through amino acid residue 4 or 6. So of course this is not going anywhere. So we've done all that, and based on the data available for paramivir co-crystallized with purified neuraminidase, we've constructed the pharmacophore model, which actually shows 7 binding points between the small molecule and the active site of the protein. What is shown here is a prediction of one of our analogs, newly synthesized interaction map with the functional groups of that molecule with amino acid residues of the active site and around of the neuraminidase. And if we compare paramivir, this is an overlay of how paramivir, which is shown in sort of gray here, it looks like yellow. That's the active conformation of paramivir, which is occupying the active site of the enzyme. And what is shown in orange is one of our analogs based on the docking study. Right? Of course, there is no structural data for this. So as you can see, it seems that both molecules are occupying the site in a very similar fashion, yet it is not identical. Right? The conformation is not identical. Again, here, this is the same active site occupancy for our compound, and this is overlay with same for zanamivir. And again, it is similar but somewhat different. So this is sort of results we would like to have to see whether those small changes we can find some which would be beneficial. So based on 7 known neuraminidase inhibitors for which there are structural data available, we've constructed the linear regression model. And when we plot predicted activity value based on this regression model, which is shown as IC50 data, when we plot it against the actual experimental data for the inhibition of the neuraminidase, we see actually a more or less nice linearity. So that suggests that if you want to be optimistic, that suggests that this model might have some predicted value. So we can take a structure of the compound and say, predict what the activity against the neuraminidase would be. So this is all nice, but not all people necessarily believe in it, myself included. So it would be interesting to see whether that would actually hold. So then we designed, synthesized, and a number of analogs of Tamiflu or zanamivir, and tested it for their ability to inhibit purified NA in vitro. And this is just to show some random compounds, some random data. So basically, their assay performs as expected. Oseltamivir gives some sort of somewhat sigmoidal curve, as well as other compounds. So we believe that assay is functioning normally. So when we now compare the predicted value, when we rank our inhibitors according to the predicted activity, which is the secondary iteration, is this E-score. When we compare it to the actual experimental data, this is what we get. So the idea is that if E-score is 15, minus 15 or lower, that would predict some decent anti-neuraminidase activity. And in generally speaking, these scores correlate pretty well with the actual IC50, experimental IC50 data. And oseltamivir, zanamivir, and peramivir show activity as expected. This is all in micromoles, right? I failed to mention that. And the best compound by experimental data is AV5080, and it actually has the best score, right? So I would say more or less with some caution, I would say that model seemed to be yielding something. And of course, there are outliers which have some scores, but essentially no activity at all, or very weak activity. So those are structures of compounds which are shown on the previous slide. So it seems that we are focusing on two side groups here and here. So then we went and tested same compounds against several strains or types. So H1N1, H5N1, and B Florida. And again, it seems that same compound 5080 scored best against all three tested strains, including, so it is active against both A and B types. Then we tested even more different isolates and and strains. And this is EC50 numbers, and this is EC90. So if you just focus on EC50, this is sort of the best way of comparing and ranking the compounds. So basically, when we look at any any strains which are Tamiflu sensitive, we are getting numbers for this compound 5080, which we selected now as sort of most interesting compound. In all those Tamiflu sensitive strains, 5080 is essentially as active as either Tamiflu or zanamivir, very similar. Sometimes a bit better, sometimes a bit worse. Also, it is active against type B, right? In type B, there is a real advantage over oseltamivir, about 10-20 times. When we look at oseltamivir-resistant strains, however, we see a striking difference, right? So oseltamivir is not active against those. Zanamivir is active, and our compound is also active. So, and that goes for all 4 oseltamivir-resistant strains here with different mutations, and also both A and B. So that made us pretty optimistic. So it seems that we've been able, through that rational drug design, we've been able to first improve the activity of the neuraminidase inhibitor in general, and more specifically, make it work against oseltamivir-resistant virus, right? That was all based on the purified neuraminidase inhibition. So when we look at the inability of those compounds to inhibit replication of virus in tissue culture in infected MDCK cells, basically that was reproduced. So again, we compare now our compound and zanamivir and oseltamivir, and we do see that our compound is actually 10-20 times better than oseltamivir in either H1N1 or H5N1. So far, so good. So would it work now in the mouse model in vivo being administered orally? So now we are comparing it in the mouse pneumonia model, infectable with a human virus adopted to propagate in mice. And we've tested several different doses from 5 milligrams per kilogram per day up to 50. And in general, here we're looking at the survival date and mean day to death. And all in all, it seems that it does work upon oral administration. And it is very similar to essentially to oseltamivir. And of course, this is an oseltamivir sensitive virus. This is just to show body weight dynamics. And this is, for example, oseltamivir, and pink is 5080, our compound. And this is, it's hard to see, 5 milligram, 10 milligram, 15 milligram. And this is a 25, 35, 40, and 50. And this is a repetition of 25. So this one is a repetition of this, right? So essentially, you can see that body weight dynamics, which reflects both activity and protective activity and potential toxicity, both compounds look essentially the same. When we plot dose dependency for versus activity, you see that this is Tamiflu, this is 5080. You see that both compounds peak, the activity peaks at 25 milligrams per kilogram per day and does not improve any further, but at optimal dose, both compounds are very comparable. So then we went and did all kind of preclinical pharmacology, starting with in vitro pharmacology, metabolism, in vitro safety, and such. So we found that this compound is binding plasma protein in relatively low, 30–35% in humans, which is not a bad thing. It has a good microsomal stability with 3 species microsomes, with or without cofactors. It is not non-mutagenic in vivo and in vitro. In vivo, in chromosomal aberration test in mice and Ames test in vitro. Her channel binding assay, it is not active, and this is predictive of cardiotoxicity. Cytochromes, liver cytochromes, we've tested all major isozymes which are engaged into metabolizing known drugs. And again, there is no inhibition up to 10 micromolar, which is the highest concentration tested. And that is predictive of low potential for drug-drug interactions in the clinic with other drugs. So this is just one example to show this is microsomal stability without cofactors or with cofactors. And this is data up to 24 hours. So we can see that in all 3 species tested, compound is pretty stable even after 24 hours of incubation with liver microsomes. So pharmacokinetics was also done in mice, rat, and dog. And this is IV on the left-hand side and peroral on the right-hand side. And just some more extensive data for the dog at 2 mg/kg IV or 10 mg/kg orally. And in general, this is a more or less okay profile, but what actually we found that bioavailability of this drug is still very, very low. Right? So it is in rat is 2.5% and in dog is 4.3%. So now the question arises, how was it efficacious in vivo, in mice, upon oral administration if their oral bioavailability is so low, right? It's a single-digit percentage. So then we tested, it is not shown here, so we've tested we tested zanamivir, which is a low bioavailability, right? And in rat, it was actually 0.5%. So at 2.5%, we have very low bioavailability, but it is still about 5 times greater than that of zanamivir. And given the fact that our compound is about 10 times more active than Tamiflu, that potentially could explain how we have comparable oral activity, efficacy for this compound to Tamiflu, while bioavailability it is much, much less than that of Tamiflu, right? So that's our current hypothesis, which we can offer. So if we'll find a way to further improve bioavailability, potentially that compound could become much more active. So toxicity in animals was undertaken. This is just to show a 6-week toxicity study in rats. So that was treatment for 30 days with 15 days follow-up, and we've tested 1550 or 150 mg/kg orally. And 150 roughly corresponds to the human equivalent, 10-time human equivalent of the predicted dose in the clinic. And as usual, everything, body weight, consumption of food, water, and everything was monitored. And then half of the animals was euthanized by 30 days, another half after their 2-week observation period. And full histopathology evaluation was also undertaken. So as a result, it is generally well tolerated and safe. Mild signs of gonodotoxicity in males was observed, And we followed up with a very extensive study of reproductive toxicity in both rats and dogs, both males and females, and we didn't find any confirmation of that. So I don't know what to make out of this here. Same was done in dogs, right? And the result was that we've seen in dogs, we've seen some mild and reversible hepato and renal toxicity at the higher dose, which is again about 10 times over projected human dose. So NOAL for dogs was 7.5, a maximum dose at which you don't see toxic effects. So this is a list of all animal tox studies undertaken, and it was pretty extensive, so that actually enables us to go into the clinic. We've planned the first-in-man clinical study, and that would be a double-blind, randomized, placebo-controlled study, and the major purpose or goal would be safety, tolerability, and PK. And we would have 4 cohorts, and we would start with 30 milligram per day, which is actually what all toxicity studies animals allow us to do, right? So with a 10-fold safety margin. Then we progressed to 80 milligrams, 160 milligrams, which would be twice a day, and 320 milligrams. So current recommended dose for Tamiflu is 80 milligram twice a day for treatment, and actually it's 75, right? So it's 150 per day for treatment for 5 days, or once a day, 75 milligrams for 10 days. So by completing that study, which would include both single dose and multiple dose, that would enable us to go into efficacy studies for both treatment and protection, right, prevention. So that study is to begin in essentially next quarter of this year. That's what we hope. And in summary, this is basically a summary of what I said. So it seems that we've been able to discover a new neuraminidase inhibitor, which is an analog of Tamiflu, which is more active, which is active in vivo, efficacious in vivo upon oral administration, which is working against Tamiflu-resistant mutants, and it appears to be safe and well tolerated in 2 rodent species and in dog. So preclinical package is completed, and we are waiting to initiate phase 1 first-in-man study. So I stop here and take questions. It's nice to see that the percentage of the audience who was asleep by the beginning of my talk did not increase. I take it as a good sign. No, we'll blame it on the transatlantic flights, of course, right?
So what are the chances of developing resistance against this drug if it is being used extensively like oseltamivir?
Pardon?
What are the chances of development of resistance against this drug if it is being used extensively like oseltamivir?
We have to assume that it is very possible, right? So far, every direct antiviral which is known to man had developed resistance this way or another, sooner or later. The question will be what is their genetic barrier to resistance, right? It could be, in other words, how quickly resistance would develop and how quickly it would spread, and what is the replication cost of the resistance. Right, so we don't know any of that.
So you cannot predict.
No, we can't. We can make lots of predictions, but I would rather not. I don't believe in those. Right? I believe in direct data. So when we generate the data, we'll be able to address this. Right now, we don't know anything. What we know is that it is active against Tamiflu resistant virus, which is, so it is not cross-resistant with Tamiflu, right?
Thank you.
Yeah.
How quickly do you see pediatric data coming out after clinical studies?
Oh. You mean was a special formulation for kids?
Not necessarily formulation, but after you do the adult studies.
So I think it would be about up to at least 2 years of the clinical studies before we can address that or begin to address that, right? Does it make sense?
Yeah. You have to make sure all the errors happen on the adults before you give the kids.
Absolutely, right.
We use things off label sometimes.
Yeah, okay, yeah. Yeah, I would not recommend it with a drug which is very new, right?
Yes, Peramivir, you mentioned in your rational design that the two drugs occupy the same pharmacophore, the same site. If Peramivir is approved, and I don't know if it is, but if it is...
Yeah, it is approved for IV.
So it
injections.
would be, resistance is likely to occur in your drug more quickly in those individuals. If lots of individuals are seeing the Peramivir, it seems to me resistance could occur in your drug too.
Not necessarily. So what I would say, we've constructed a pharmacophore, and based on their docking studies, our inhibitors are occupying obviously the same active site, and we can assume they work through the same molecular mechanism, but they are not overlapping directly with any of their either Tamiflu or Peramivir or Zanamivir. They are they're yet different from all of those, right? So we can't say it is more direct analog of Peramivir. We used Peramivir data to begin with, but it doesn't mean that our compounds are closer to Peramivir. In fact, they are not. I think based on that cross-resistance data, they are closer to Zanamivir in their resistance profile, right?
Any more questions before we break? Excellent. Well, thanks very much to our speakers this morning for keeping to time. And